This mechanism of toxicity has four steps. So first two steps will were covered already in the first session. That is delivery to the target and the reaction of the ultimate toxicant with the target molecule. So I just want to give a brief on this too as well. So this target delivery to the target of the chemical has some steps in that, which is absorption, metabolism, distribution, and elimination, where that helps the target molecule to deliver at the site where it produces the toxicity. And the second step reaction, and after delivering to the target site, the molecule will undergo some reactions and it forms a relevant molecule, and instead of the drug it goes into the bloodstream, and it shows its effects by different ways, like means formation of nucleophiles, free radicals, oxidants. After forming all these things, it shows its effects, it shows its toxicity. So coming to the third step, so this is a brief of two steps. Now coming to the third step, after attaching the molecule to the after attaching the chemical to the target molecule, what kind of effects it shows, that is the third step. And fourth step is what could be the repair mechanism from our cell side, and what could be the adaptation mechanisms. That two steps we are going to I'm going to share in this session. So yeah, so as I said, this is the third step, where cellular dysfunction and resultant toxicities. So generally, toxicity induced cellular dysfunction, that is completely depends on the function of the target molecule, because based on the target molecule function, the toxicity will get induced by the chemical. For example, if the function of the target molecule is involved in cellular signaling and regulation, then the toxicant alters a gene expression present in the alter the gene expression of the target molecule, and also it affects the cellular function as well, alter the cellular function. So if the function of the target molecule is involved in cellular signaling and cellular function, then it alters the functions of the target molecule. And if the target molecule has a function of essential, like cellular essential process, that results in the dysfunction of that process, and that leads to the death of the cell, or that leads to the injured that injures the cell. So this is a pictorial representation of the process of the step 3, where the blue indicates the cellular regulation and the target molecule function, and the pink indicates the effect of the chemical on the target molecule. For example, the role of the target molecule is on cellular regulation. Then what happens if the chemical comes and affect the target molecule? It alters the gene expression as well as the impair the cellular function. So if it alters the gene expression, that leads to the inappropriate cell division, leads to neoplasia and teratogenesis, and also apoptosis, which is tissue involution and teratogenesis. Inappropriate mechanism of this, like an inappropriate protein synthesis, that leads to the toxicity. And the example for this is peroxisome proliferation. And now if the chemical comes and binds to the target molecule and impairs the cellular function, then that leads to inappropriate neuromuscular activity, that is tremor, convulsions, spasms, cardiac arrhythmia. All this occurs when if the target molecule function is this. Then coming to the cell maintenance, if the target molecule role is in the cell maintenance, then that if the chemical comes and binds to the target molecule, that leads to the impaired internal maintenance of the cell, that leads to the impaired ATP synthesis in the cell, and calcium regulation in the cell, and protein synthesis, and microtubular function, and as well as membrane function in the cell. All these impaired functions leads to cell injury or death. And next one, if impaired external maintenance. So now the above one which we discussed, that is completely internal part of the cell. Now if the impairment of the cell impairment is on the cell external part of the cell, that leads to hemostasis and leads to bleeding. And coming to the toxicity, toxicant induced oh sorry toxicant induced cell dysregulation. So this has a transcriptional process as well as the numerous pathways which are involved in signal transduction cascades that contribute to the toxicity-induced alterations in cell regulation and gene expression. There will be many cell receptors which transmits the cells signals to the regulatory regions of the genes which are present in the cell, and that leads to the functional proteins. So this is if there is any impairment or if there is any alteration in this pathway, that leads to the that increases or decreases the protein expression by the gene, that impairs the cellular process, and that effect that leads to the toxicity. And gene expression is also a process where the gene gets synthesized from the functional gene product, and that genetic information may be transcribed from DNA into messenger RNA, which in turn translated to proteins, and these proteins will be like that could be replicated in the multiple cells. So this is the gene expression process. So if there is any impairment or if there is any obstruction in this process, then that leads to the toxicity. Then the non-coding RNAs, like there are many non-coding RNAs like small interfering double-stranded RNA (sRNA) and miRNA. So these two are the RNAs where mostly toxicity mechanism happens, because not the siRNA also, especially the miRNA, which is a microRNA, where where it's a very short sequences, 20 to 25 base pairs will be there, and it is derived from the RNA transcripts, and that has a foldback and that it forms a hairpins, where siRNA is generated from a longer regions of double-stranded RNA, and these two from these two also mostly miRNA has more toxicity mechanisms where the chemicals come and bind to this, and that leads to the toxicity. And with the chemicals which alter the gene expression, that affects the mRNA and miRNA, as I said earlier, or it might affect the both RNAs, and that alter the gene expression by affecting directly the transcription. The transcription part, that could affect the transcription, or that could affect the signal transduction pathways, or that could also affect the synthesis of the gene or storage or release of the extracellular signaling molecules. By impairing all these or any of these mechanisms, that can lead to the toxicity of the cell or injury of the cell and or injury of the gene. Coming to the dysregulation of transcription. First, gene transcription. What is meant by gene transcription? It's a control largely, it is gene transcription is controlled largely by interplay between transcription factors and the regulatory or promoter region of genes coding for mRNA and miRNA. So by binding to these distinctive nucleotide sequences in the promoter, ligand and signal activated transcription factors, all these facilitate and forms a pre-initiation complex that increases the repression transcription, that increases the transcription, and that continues with the replication, and this is a general process of the gene transcription. But if any xenobiotic or if any chemical come and interacts the transcription factors and also the pre-initiation complex which was formed by the transcription factors, that alter the promoter region of the gene, that leads to the toxicity. Example for these are hormones and vitamins, retinoids and vitamin D, that influence the gene expression by binding to the gene or by activating its transcription factors or intracellular receptors. So mostly this is one mechanism, and second thing is mostly that there will be some chemicals which directly mimic the natural ligands, and when they go and bind to the receptors or on the gene, or they affect the gene directly, and it reduces the effect of the normal drug or chemical normal process, and that leads to toxicity. So example for this gene transcription inhibition or gene transcription impairment is clofebrate and other fibric acid lipid lowering drugs and phthalate esters that also act as substitutes for polyunsaturated fatty acids as ligands for the peroxisome proliferator activated receptors. And the other example is estrogens, which is DES, DDT, bisphenol A. What they act as endogenous ligands for endogenous estrogen receptors. They go and bind to the estrogen receptors rather than the estrogen hormone. So that leads to toxicity. Second one is ligand activated transcription factors. So these transcription factors have control many fundamental cellular process where adverse effects of the compounds act through the transcription factors and result the disturbance of cellular proliferation or by inducing the cell death. The examples for this ligand activated transcription factors is glucocorticoid receptor agonist dexamethasone that induces the apoptosis of lymphoid cells. Though this is a desirable in the treatment, but that leads to the lymphoid malignancies. That malignancies has a toxicity effect. Another TCDD produces thymic atrophy by inducing thymocyte apoptosis. And the third one is transcription factor mediated toxicity, where this is defined as mechanistically genetically modified mouse models. This is and mice overexpressing estrogen receptor alpha, ER alpha, are very sensitive to the DES induced uterine adenocarcinomas, whereas ER alpha knockout mice are resistant, demonstrating a role for ER alpha in tumorigenesis. Similarly, the mitogenic and hepatic tumor promoting effects of peroxisome proliferators in rodents were conformed to the receptor mediated because they are not observed in the PPAR alpha null mice. The compounds that act on ligand activated TFs also evoke changes in the metabolism of chemicals by inducing overexpression of relevant metabolizing enzymes. For example, PPAR alpha ligands induce lipoprotein lipase and enzymes involved in fatty acid oxidation. And finally the transcriptional regulation, where the direct engagement with response elements in in the promoter regions of the gene, direct engagement of the chemical will be there instead of the transcription factor engagement. That leads to the leads to the binding of the chemical or the toxicant to the gene, and that leads to the toxicity. After TC, like example is TCD that binds to AHR complex with the nuclear translocator protein, and the complex bind to dioxin response element DRE or XRE to that initiates the transcription. Coming to the transcriptional dysregulation by chemical alteration of regulatory region of genes. There is a region of there is a specific region on genes. If any toxicant or chemical comes and act on the specific region of the gene, that is called as regulatory region of the genes. So in like as I said, xenobiotics may alter the regulatory regions, though through direct chemical interaction. Either there could be by a direct chemical interaction or by changing the methylation patterns of the genes. The good example of this is thalidomide that causes developmental defects by coming and intercalating at the GC boxes. There is a specific area of the gene and binds. Thalidomide comes and binds to that particular box, and that leads and the transcription factors like specificity protein 1 and early growth response protein 1, all these will be like despaired or impaired, all these factors. Instead of these factors binding to that boxes, thalidomide goes and bind to the boxes and form and shows the developmental toxicity. Like as I said, generally the angiogenesis or normal limb development should happen by using the growth factor IGF-1 or fibroblast growth factor, which happens. This is a specific time where the embryonic development happens. In that case, in that time, these genes will act, and this helps in production of angiogenesis or limb development. In this phase, if thalidomide goes and binds to the specific boxes of the gene, which intercalates with that boxes and inhibits the growth factors or these fibroblast growth factors, that leads to the developmental toxicity. And methylation of cytosine bases in CpG islands in the promoter regions of gene, that is also a major epigenetic mechanism where that is also coupled with histone modification, and that influences the histone and modifies the histone part of the gene. This epigenetic mechanism is one that affects the heritable traits. Like the heritable traits means the genes which are coming from our ancestors, that if that genes has been affect, then that could be that is called as epigenetic mechanism. So this happens by promoter methylation. If the methylation is more, if the methylation got increased at the promoter phase, then that silence the CpG, that silence the epigenetic mechanism, or that silence the gene. Or if the promoter methylation is decreased, then that leads to the decrease of if there is a decrease in the promoter methylation, that shows the aggressive effect on the gene, where that gene will get aggressive, and that shows the toxicity. Importantly, when DNA replication occurs, this methylation pattern Where it is helpful means as I said, these are heritable genes, right? So if these methylation patterns will affect, uh, they are useful for the DNA replications. So if any drug or chemical comes and affect this methylation pattern, then that inhibits the DNA replication or modifies the DNA replication and forms a toxicity in the genetic material, that is which is copied from the strand and daughter strand by DNA methyl transferase 1. That pattern comes as a hereditary genes. And coming to the role of miRNAs in toxicant induced alteration in gene expression. As I said earlier, most of the chemicals of the drugs will come and act on the miRNAs, because they repress the translation or increase mRNA degradation to decrease gene and protein expression. So any toxicant that increases miRNA activity, that decreases gene expression, and if any toxicant decreases miRNA activity, that increases the gene expression. These small non-coding RNAs, which are derived through a multi-step process, or overall they regulate many fundamental cellular functions, resulting in a broad range of potential adverse effects. So as I said, this miRNA is a RNA where most of the chemicals come and affect. They are as I said, they are very nearly 2,500 mature miRNAs that have been identified in the human genome. There will be 2,500 mature miRNAs, and that miRNAs contribute for many mechanism of toxicities. So that so for example, if miRNA which is in liver, that is MIR 122, that comprise about 50% and 70% of the miRNA in adult mouse and human liver. So that miRNA 122, that is regulated by the liver enriched transcription factors, or including hepatocytes and nuclear factor 6 and nuclear factor 4A, which is the program that controls that program controls proliferation, differentiation of both hepatocytes and biliary epithelial cells. So if this miRNA 120 is also implicated to have a central role in development of fatty liver cirrhosis, because genes involved in lipid biosynthesis are upregulated in MIR 122 null mice. So here is the pictorial representation of biogenesis of microRNAs, where the miRNA gene undergoes breaks in the nucleus. It breaks by using an RNA polymerase and forms a prime micro RNA, which is a much longer compared to miRNA. And this prime micro RNA in the nucleus itself, with the help of RNAs DROSHA and DGCRS, that undergoes cleavage and forms pre-micro RNA. This pre-micro RNA from nucleus, it transports to it transports to the cytoplasm by using an exportin 5 enzyme, and after coming to the cytoplasm, this miRNA gets cleavage with the help of dicer and TRBP, and this miRNA with the help of protein AGO2 forms a intelligent forms a complex and RASC formation that leads to the mature microRNA. So coming to the next thing, dysregulation of signal transduction. So signal transduction is a process by which a chemical or physical signal is transmitted through a cell in a stepwise of events. So after involving kinase or phosphatase activity, that modifies protein activity by phosphorylation status to invokes a response. So as such, many extracellular signaling molecules that includes growth factors, cytokines, hormones, neurotransmitters, and some secreted proteins, all these engage intracellular signal transducing networks, and ultimately that activates the transcription factors. As the major signal transduction pathway is the mitogen activated protein kinase MAPK pathway, and that is also referred to as extracellular signal regulated kinase. And the signaling in this pathway is initiated by binding of a signaling molecule. One molecule comes and binds to the cell surface receptor, such as like EGF, epidermal growth factor, and after binding to that receives the ligand that receives that binding, and it shows it undergoes phosphorylation. This figure 7 represents this MAP kinase pathway, where, as I said, ligand comes and binds to the epidermal EGFR factor, growth factor receptor, where this, after binding to this, this RAS receptor activates by converting from GDP to RAS GTP, and after activating it forms MAP3K, MAP2K, and MAPK. And after that, this MAPK goes to the nucleus, enters to the nucleus, and it undergoes the transcription factors. It binds to the transcription factors and undergoes the different mechanisms from there. And this is the figure 8 is the signal transduction pathways which are involved by phosphorylation and potential interactions and across every all the pathways which undergoes phosphorylation are included in this figure, where this 127 in this figure 127 numbering represents the symbols of cell membrane receptors along with activating ligands as well. And here, as I said, EGFR, this mechanism is also there in this uh, pathway. Like this pathway is also mentioned. The number 4 pathway is the EGFR MAPK signal transaction pathways. So in this pathway, there, as I said, there are 7 cell membrane receptors. Which first receptor is a JAK-STAT pathway, and second and third is NF alpha beta activation pathway, and fourth, as I said, it's MAPK pathway, and fifth one is integrin receptor mediated activation of the focal adhesion kinase pathway, and sixth one is the PKC pathway, the phospholipase C activation, and seventh one is hormone mediated activation of CAMP and protein kinase APK pathway. All these pathways after binding, if ligand comes and binds to that, that undergoes phosphorylation and shows its effect in the cellular membrane, and that signal activated transcription factors that release the signal activated transcription factors which helps in cell cycle progression, mitosis or mitosis. All these process leads to the mitosis and cell proliferations. So if any chemical comes and binds on these receptors instead of the ligands, then that leads to the cellular disruption or cellular dysfunction, and that leads to the toxicity and chemically altered signal transduction and proliferation. So here this particular step facilitates in phosphorylation, where that promotes mitosis, as we saw in the earlier slide. The all the phosphorylation pathways leads to mitosis and tumor mitosis, and that leads to the tumor formation. So examples include Forbol esters and fumarin B that activate protein kinase, which is the sixth pathway in the earlier slide, that activates PKC, promotes mitogenic activity by phosphorylation of RAF and by phosphorylation of a protein phosphatase that dephosphorylates the TFC junction at specific sites, thereby permitting its binding to DNA. That leads to the DNA binding. And chemicals that modulates PKC activity often mimic the diacyl glycerol, that is an endogenous activator of PKC, and the other PKC activator is calcium, which can be mimicked by lead. So here this lead, in contrast, this lead has a two different mechanism on the same pathway. Like if lead ion has a if it is in a picomolar concentration, then that increase the PKC activity. If that lead ion concentration is high and if it is in micromolar concentration, that inhibits the PKC activity. The next step, chemically altered signal transduction with anti-proliferative effect. So here cell proliferation generally that has a tight control then that would be turned off when repair of injured cells is completed. For example, if any cells got injured, in that case the cell proliferation starts, and that cell proliferation will that has to be stopped tightly after the recovery of the cells. But however, in this process, if there is a decrease in proliferative signaling cell injury, then that leads to then the replacement of the injured cells will be compromised, and that can direct a cell to the apoptic death. The example for this is PKC inhibitors, staurosporin, and P1K inhibitors, workmanin, and IEKB degradation gliotoxin that induce apoptosis, and TGF-B and glucocorticoid increase IKB, which in turn decrease NF activation and CMC expression. And both mechanisms contribute to the apoptic effect of TGF beta and glucocorticoid, that particularly seen in the lymphoid cells. Coming to the dysregulation of extracellular signal production, here the extracellular, this activity completely happens on the surface of the cells. So mostly hormones are part of this. Hormones will the chemicals will affect the hormones of anterior pituitary that exert mitogenic effects on endocrine organs by acting on cell surface receptors. So pituitary hormone production generally would be negative feedback where peripheral organs and disturbance of the circuit adversely affects pituitary hormone secretion and endocrine organ function. So if the decrease in the pituitary hormone secretion happens, then that leads to the endocrine organ dysfunction. Example to this is thyroid hormone production, which is that enhance the like if any chemical inhibits the thyroid hormone production, that enhance the example that leads to the herbicide. The example for this is herbicide amitrol in the fungicide metabolite. And if there is any enhancement in the thyroid hormone elimination, phenobarbital, that reduces thyroid hormone levels and increases secretion of TSH hormone, and that reduces the feedback inhibition. The increased TSH secretion stimulates the cell division in the thyroid gland, that leads to goiters and thyroid tumors as well. And decreasing the secretion of pituitary hormone produces the opposite adverse effects, that leads to apoptosis followed by involution of the target organ. Example for this is estrogens produce testicular atrophy in males by means of feedback inhibition of gonadotropin secretion. Coming to this impaired ongoing cellular activity, where the ongoing control of the specialized cells is exerted by signaling molecules which are acting on the membrane receptors that transduce the signal by regulating the calcium entry into the cells, that also stimulates the enzymatic formation of intracellular second messengers. So these alters the phosphorylation of functional proteins and changing the activity of the proteins. Uh, so the steps included in this mechanism are neurotransmitter concentration. Based on the neurotransmitter concentration or differences in the concentration of the neurotransmitter, this particular ongoing cellular activity might get impaired. Or the receptor function of receptor, the alteration in the function of receptor happens by binding to the chemical, then that also leads to the impaired ongoing cellular activity. And intracellular signal transduction and signal terminating process. So coming to the dysregulation of electrically excitable cells, where this influence the cellular activity in excitable cells like neurons and skeletal, cardiac, and smooth muscle cells. The transmitters and the modulator, which are generated from the neurons, that control the cellular functions like release of neurotransmitter and muscle contractions. So if these undergoes the dysregulation, that leads to the toxicity. The main example for this is the tetrodotoxin that blocks the voltage gated sodium channels, that is in the motor neurons, but the observed effect is skeletal muscle paralysis. And cyclodine insecticides, this is also one of the good example that blocks GABA receptors in CNS, induce neuronal excitation and convulsions, and alterations in the neurotransmitter levels. So for example, if any chemicals which that has a capacity to alter the neurotransmitter levels in the in the synapses by interfering with their synthesis of the neurotransmitter, or by the storage of the neurotransmitter, or by the release or removal of the neurotransmitter, that leads to the uh, dysfunctioning of the physiological activity of the neurons, and that leads to the toxicity. The main example for this is the decrease in the GABA synthesis that happens by hydrazine and isoniazid drugs, that causes convulsions like that. And setpin, this is also one of the that also inhibits the neuronal storage of norepinephrine, 5-hydroxytryptamine, dopamine, that leads to the toxicity. And botulinum toxin, that has a capacity of skeletal muscle paralysis, that also inhibits the acetylcholine release from motor neurons, preventing the stimulation. That affects the adverse effect, that has a adverse effects by inhibiting this acetylcholinesterase. Then toxicant neurotransmitter receptor interaction. Generally, the toxicant comes and binds to the neurotransmitter receptor rather than the ligand or the drug comes and binding and show its effect. There are different types in this as well. Agonists that associate the ligand binding site on the receptor and mimic the natural ligand. The chemical also mimic the natural ligand, comes and binds to the receptor. Those are called as agonists. And antagonists are the one which comes and bind to the receptor and show the opposite effect. That does not activate the receptor, so that there won't be any further process down the line to further receptor happens. And so third thing is activator. The chemical comes and binds to the receptor and it activates it, and the increase of the that multiplies the process, so that that leads to the adverse effects or toxicity. And inhibitor, when the chemical comes and bind to the receptor, and that inhibits the further steps. And the example for this is mucimol, that is a mushroom poison that acts as an agonist to the inhibitory GABA A receptor, whereas barbiturates, benzodiazepine, general anesthetic, and alcohols that activates the GABA A receptor. Sensory neurons have membrane receptors that are stimulated by noxious chemicals and operate as ligand gated cation channels. The transient receptor potential TRP channels open to enable sodium and calcium influx. Here the TRPV1 is stimulated by capsaicin, and that leads to the pungent that leads to the irritating effect. If capsaicin comes and activate to this receptor, that shows the irritating effect. Or and if any hot pepper hot pepper comes comes in like that, if we expose to any hot pepper, these receptor will get activated, and that shows is saying that okay, burning sensation or corrosion happens in the capsaicin, and burning sensation or irritating sensation happens by activating this receptor. However, TRPA1 receptor that is activated by thiol reactive and oxidant chemicals such as lacrimatory compounds. If we expose to tear gas or methyl associated and corrosive gases, this particular receptor will get activated, and that leads to the secretion of lacrimation. If we expose to tear gas, secretions happens by activating this receptor. Yeah, and toxic and signal transducer interaction. Many chemicals alter the neuronal and muscle activity by activating on the signal transduction process. So voltage gated sodium channels that transduce and amplify excitatory signals, which are generated by ligand gated cation channels, and that also get activated by a number of toxins as well, which derive from plants and animals as well as some synthetic chemicals. The examples for those is DDT, and that results DDT results to the DDT comes and binds to this, or it shows its signal transduction that leads to the overexcitation. In contrast, chemicals that block the sodium channel blockers, sodium channel, so that after blocking it leads to the paralysis. So example for this is as we saw earlier, tetrodotoxin and saxitoxin that binds to, that blocks the sodium channels, that leads to the which this sodium channel helps in signal transduction in the sensory neurons, that blocks the signal transduction for the sensory neurons. Toxicant that has a signal terminator interactions. The cellular signal generated by cation influx is terminated by the removal of the cations through the channels or by the transporters. Example for this is arachidonic acid metabolite, which is 20 hydroxy 58 11 14 to 20 HTE, that blocks calcium activated sodium channel potassium channels, causing the vasoconstriction. In fact, the induction of 20 HTE production by cyclosporin in renal proximal tubules and the resultant decrease in renal corticate blood flow may contribute to the nephrotoxicity of the cyclosporin. And the blockade of other example is the blockade of HERG channels, that is a voltage gated potassium channels, that causes prolonged delays in the cardiac repolarization, as indicated by prolonged QT time in the ECG. And coming to the next, dysregulation of activity of other cells. While many signaling mechanisms also operate in non-excitable cells, but usually these are less consequential because dysregulation happens. This process is this happens in very less. There won't be any consequences if this process happens also. The example is rat liver cells that possess alpha-1 adrenergic receptors, whose activation increases the metabolic changes or disrupt the metabolic changes like increased glycogenolysis and glutathione export by elevating the elevating the intracellular calcium ions. And toxic alteration of cellular maintenance. The major toxicity in the cellular maintenance is cell death, that which is a irreversible process. We can't reverse that process because we can't reverse the cell death. So that can also disrupt the structural and functional integrity of the organ or the organism, because if any cell death happens, that could affect the organ or that particular organ or the organism, and this invokes the numerous repair or adaptive process to compensate for the loss of function. And coming to the mechanism of toxic cell death, there are three common major events which underlie sorry, there are three major events that underlie for this cell death. Those are ATP depletion, sustained rise in intracellular calcium, and overproduction of ROS and RNS. These three mechanisms leads to cell death. So cell death was considered to occur by, generally it happens by necrosis, that is a process deemed to irreversible damage of the cell. These pathways primarily differentiate by the agents that induce the effects and the mediators of the cell death. So the agents which comes and induce the effects that mediates the cell death, these agents will come and affect on that mediators and leads to cell death. Generally it happens in the mitochondria because mitochondria forms a core of every cell death regardless of the pathways or the mediators involved. And first step is primary metabolic disorders jeopardizing cell survival. Generally in this cell viability, ATP, as I said, ATP synthesis, which is helpful in most of the process in the cell, that helps in cell viability. It supports intermediary metabolism and also has a major source of cellular energy. That ATP is utilized in many process, many reactions or activating endogenous ligands or by phosphorylations and adenylation, and it is incorporated into cofactors as well as nucleic acids. So ATP is useful in many many processes or many steps that drives many different ion transporters as well, that helps in driving the ion transporters, sodium potassium ATPase in the plasma membrane, calcium ATPase in plasma, ER membranes. All these membranes work with this ATP only. The main example for this ATP depletion is sodium concentration gradient across the plasma membrane, which is generated by sodium potassium pump that drives sodium glucose and sodium amino acid co-transporters as well as sodium calcium antiporter that facilitates the entry of these nutrients and the removal of the calcium. This is a major example of ATP synthesis. And second one, sustained rise of intracellular calcium. Intracellular calcium levels are highly regulated, and these are very 10,000 fold difference between extracellular and cytosolic calcium concentrations. It is maintained by the impermeability of the plasma membrane to calcium and by transport mechanism that remove calcium from the cytoplasm. So mainly mitochondria express a low affinity to calcium transporter that plays a significant role in calcium sequestration when the cytoplasmic concentration rise into the micromolar range. So under these conditions, a large amount of calcium accumulates in the mitochondria where it is deposited as a calcium phosphate. So example for this is the similar the toxicant may increase cytosolic calcium by causing it to leak from the mitochondria or the endoplasmic reticulum into the cytoplasm. So from mitochondria, the calcium goes into the cytoplasm and it increase the concentration of the calcium in the cytoplasm will get increased, and they also diminish the calcium efflux through inhibition of calcium transporters or depletion of their driving forces. So the main example is glutamate receptor agonist. That is a ligand gated glutamate kinase domain where that this particular glutamate comes and binds to the calcium transporter and that inhibits the calcium transporter channel, so that there is an increase in the calcium in cytoplasm, which leads to the cell death. Overproduction of ROS and RNS. This ROS and RNS is directly generated to that cause redox cycling and transition metals. In this overproduction of ROS and RNS can be secondary to intracellular hypercalcemia, as calcium activates enzymes that generate ROS and RNS by activating dehydrogenases in the citric acid cycle, which accelerates hydrogen output and in turn increases the flux of electrons along the electron transport chain. So the main example for this is cyanide toxicity, which is in neurons that is associated with rapid depolarization and glutamate release, that is followed by calcium influx through voltage gated as well as glutamate gated channels. Mitochondrial permeability transition and necrotic cell death. So formation of non-specific pore that opens when mitochondrial calcium levels are high, resulting in generation of ROS and RNS and depletion of the ATP. That leads to the depletion of the ATP, so that the cell undergoes necrosis and death, and finally it leads to death. So collectively these events produce the biochemical, if this happens in the cell, that leads to the biochemical dysfunctioning and mitochondrial swelling, because increase in the calcium levels in the mitochondria leads to the swelling of mitochondria and cell death. So calcium in the mitochondrial matrix effluxes out of mitochondria through the pore to increase cytoplasmic calcium concentration. So this pore formation from mitochondria, it goes to the cytoplasm, and there the calcium will get accumulated. Other mechanism of cell death. Although necrotic cell death is common mechanism by which toxicant, mostly toxicant acts on this necrotic cell death only, but in the case of necrosis, the common steps outlined above occur in a somewhat uncontrolled cascade. So they form cell death typically involve a program or more ordered cascade that ultimately triggers a irreversible damage. These programs includes apoptosis, necroptosis, and ferroptosis, and autophagy, and the general morphologic, biochemical, and molecular features of these pathways. And coming to the apoptosis is a mechanism which is characterized by the pycnotic chain, cytoplasmic shrinkage, nuclear fragmentation, and blebbing of cell membranes. And ultimately, apoptotic cells disintegrate into membrane enclosed fragments called apoptic bodies. Like necrosis, mitochondrial dysfunction, and MPT contributes to apoptosis. So necroptosis means this is another form of programmed cell death, which is an inflammation related cell death. So here in this program form, necrosis triggers by ligands of the death receptor family, and its development is regulated by receptor interaction, serine threonine protein kinase 1 and 3, and the mixed linase kinase domain like protein MLKL. So this with these proteins, the necroptosis occurs, and necroptosis is distinguished from necrosis in that is a programmed process, and although the cell membrane is disrupted, this process is also tightly regulated. Then coming to the ferroptosis, this is another recognized form of regulated cell death. This is a process characterized by the accumulation of lipid peroxidation products and ROAs that are derived specifically from iron metabolism. So ferroptosis is recognized as a non-apoptic form of cell death, in which the toxicant that inhibits glutathione biosynthesis or the glutathione dependent antioxidant enzyme glutathione peroxidase are integrally involved in this mechanism. So induction of cell death by unknown mechanisms. So in this process, there won't be any mechanism involved, like as we saw in the earlier steps. In this the mechanism is unknown. This is just the damaging of the plasma membrane. The cell death happens other than necrosis or programmed cell death pathways. If any other pathway other than this necrosis or programmed cell death pathway, then that step includes in this, that process includes in these unknown mechanisms. So chemicals which directly, example for this mechanism is chemicals that damage the plasma membrane, like solvents, detergents, and venoms which are derived by hydrolytic enzymes that cause direct cell rupture and leads to death. So there are some lysosomal membrane lead to tissue degradation by release of lysosomal hydrolases. Example for this is aminoglycoside antibiotic and hydrocarbon binding to alpha 2 globulin that injure the renal tubule cells. Other mechanisms of cell death include destruction of the cytoskeleton also, such as observed with microfilament toxins, perandoin and cytocalicins. So coming to the step 4, inappropriate repair and adaptation. So this step is a progression of toxic lesions that can be intercepted by repair mechanisms operating at molecular level, cellular level, and tissue level. So these cells and organisms also adapt to the toxicity and renders them more resistant to anticipated adverse effects. So for example, this adaptation mechanism is one where the body or cell will adapt to the toxicity, and that reduces the toxicity by adapting to the toxic response. And here is the representation of this step 4, where this repair happens by in the molecular level, cellular level, and tissue level. In the molecular level, protein repair happens, lipid repair happens, and DNA repair happens in the molecular level. In the cellular level, the repair happens by using the autophagy of damaged cell organelles or regeneration of the damaged organs, axons. Yeah, coming to the third one is tissue level repair, where the repair happens by apoptosis, cell reproduction, or ECM reproduction. And coming to the mechanisms of repair, some chemical alterations such as oxidation of protein thiols and methylation of DNA, that process will get simply reversed, so that the mechanism of repair happens by using that processes. So hydrolytic removal of the damaged unit, if there is any unit which got damaged, that will be removed hydrolytically, and that is newly synthesized and after correcting unit often occurs with chemically altered DNA or protein and peroxidized lipids. So in some instances, the damaged molecule is totally degraded and resynthesized. This process takes much time, but in unavoidable cases such as regeneration of cholinesterase after organophosphate intoxication, that happens. That is an example of this. So repair of protein by using thiol groups. So thiol groups, which are essential for function of many proteins, that undergoes oxidation and forms protein disulfates, protein glutathione, mixed disulfates, and protein sulfonic acids. That is reversed by reduction and thioredoxins and glutoredoxins. And small proteins that feature active cysteine residues are the major endogenous protein reducing enzymes. And there are two isoenzymes of these proteins, which are thioreductin reductase, that is distinguished by intracellular location. Those are cytosolic and mitochondrial. So when these thiol groups undergo catalytic these thiol groups in these proteins are the catalytic oxidation, they reduce they undergo reduction by NADPH generated by associated dehydrogenase and glucose 6 phosphate dehydrogenase and 6 phosphogluconate dehydrogenase. ROS may also oxidize methanins in proteins into sulfoxides, forms both S and R epimers that can be reduced by methionine sulfoxide reductase A and B enzymes. So coming to the repair of proteins, which are heat shock chaperones. So these heat shock chaperones are soluble intracellular proteins such as cytosolic enzymes, which are typically folded into a globular form with hydrophobic amino acid residues folded internally and hydrophilic residues located externally and a ligand binding site that resides in a hydrophobic cleft. Like if you see here in the picture representation here, the folded protein that has a hydrophobic amino acid residue internally and hydrophilic residue located externally, and undergoes and then it forms a misfolded unfolded like folded protein, and physical or chemical insults may evoke an unduly large opening of this cleft, which lead to the protein denaturation and aggregation. So molecular chaperones such as heat shock proteins HSP 90, 70 and HSP 40 that prevent this protein folding by clamping down onto the exposed hydrophobic region of their client protein. So this exposed region they clamp down and they protect the protein. That's why they repair the proteins. HSP 90 is critical for stabilizing the cleft and impeding further unfolding, which would eventually lead to the degradation of the protein by the ubiquitin proteasome system. This function can be blocked by the compounds such as glendamycin and herbimycin that promote degradation of HSP 90 bound proteins by the UPS. So if you see here in this picture, so here this protein comes and like it activates the ATP where it forms the ubiquitin activation and it leads to the ubiquitin conjugation, and the third step is ubiquitin ligase and it forms a protein, and finally it forms a proteasome, and this proteasome undergoes deconjugation and forms free ubiquitins. Yeah. So coming to the repairs of lipids, phospholipids which contains fatty acids, hyperoxides are generally hydrolyzed by phospholipase A2 with the peroxidase fatty acids which are replaced by the normal fatty acids. These peroxidase lipids, example glutathione peroxidase, glutathione reductase system, they may be reduced. So these peroxidase lipid may be reduced by this glutathione peroxidase and glutathione reductase, and they repair the lipids. The NADPH is needed to repair the reductants that are oxidized in the process. And coming to the DNA repair, the damaged DNA repairing the damaged DNA, that is inevitable as it is spontaneously as it occurs spontaneously during replication, and it's frequently targeted by electrophiles and the free radicals. So the nuclear DNA is stable actually in part because it is packaged tightly in the chromatin, and because complex repair mechanisms have evolved to correct the adverse alterations. But coming to the mitochondrial DNA is very prone to damage because there won't be any that such kind of mechanism where chromatin and complex repair mechanisms won't be there on this. So mitochondrial DNA is prone to the damage. That damage repair mechanisms are central to toxicity culminating in carcinogenic and mutagenic effects. This DNA repair mechanisms or the disruption of normal DNA repair mechanism remain an important aspect of understanding general mechanism of toxicity, and these are summarized in this way: like direct repair, directly certain covalent DNA modifications are directly reversed by the enzymes. So DNA phosphotolase which directly repair the enzymes and cleaves the adjacent pyridines dimerized by the UV light. The enzyme uses energy of visible light to correct damage, so its function is restricted to light exposed cells. So minor DNA adducts such as methyl groups that are attached to the O6 position of guanine that are cleaved by the O6 methyl guanine DNA methyl transferase. The example for this is while repairing the DNA, this alkyl transfer resin activates itself by transferring the adduct onto one of its cysteine residues, after which the enzyme is subjected to ubiquitination.
We can't hear you.
Sir, is everything all right, sir? Hello, Pradeep sir.
Voice is
Yeah, can
not
you?
audible.
Hello, hello.
Is everything all right, sir?
Yes, yes. Can you hear me?
Okay. Yeah, yeah, we can hear you, sir. Yes.
Yeah, yeah, yeah. So yeah, coming to the base excision lesions, these lesions do not distort the helix typically, and they're removed by the base excisions, in which the altered base is recognized by the DNA glycosylate that hydrolyzes the N glycosylic bond that releases a modified base and creating an apurinic or apurinic site in the DNA. So example for this is 8-hydroxyguanosine, which is a major mutagenic product of oxidative stress and also removed from the DNA by a specific DNA glycolase. And as I said, the AP site is recognized by the AP endonuclease enzyme, which hydrolyzes the phosphodiester bond adjacent to the basic site. After it's removed, the basic sugar is replaced with the correct nucleotide by a DNA polymerase and is sealed in place by a DNA ligase. And non-homologous end joining, where this process repairs double-stranded breaks that may be formed when two SSBs occurs in close proximity or when DNA with SSBs undergo replication. This repair system directly ligates the broken ends without the need for a homologous templates. So these DSPs are recognized by the KU protein. So example for this is KU proteins KU70 and KU80, and then that binds and activates the DNA dependent protein kinase catalytic subunits and undergoes a mechanism, repair mechanism, and recombination or repair. This this used to repair post replication gaps and DSBs and double stranded breaks. So post replication gaps form when that forms when a bulky adduct or an intra-stranded pyrimidine dimer does not occur before DNA replication. So at replication, two homologous dissimilar DNA duplexes will be formed, which have one has a large post-replication gap in its daughter strand and an intact duplex synthesized at the opposite leg of the replication fork. So this intact duplex is utilized to complete the post-replication gap in the damaged duplex, which is accomplished by the recombination of the appropriate strands of the two homologous duplexes. So the main example of this mechanism is proliferating cell nuclear antigen, which is a central player in post-replication repair. And this PCNA also binds to chromatin and slides along the DNA strand to tether the polymerase to the DNA template. And this PCNA is a platform for a wide variety of proteins that participate in DNA replication and damage. And coming to the cellular repair, this is a repair mechanism where or this mechanism will has the ability to repair or remove the damaged cell organelles. The main step or process included in this is autophagy, which autophagic removal. This happens by autophagic removal by like by engulfing the damaged cell organelle or by removing the damaged cell organelle. And this also may be this is the universal mechanism of cellular repair, where the clearance and regeneration of the damaged axons in a mechanism specific for nerve cells. So autophagy is important, as I said, autophagy is an important mechanism for removing misfolded proteins from cells that resulting from oxidative stress or ER stress. So ER stress that is a adaptive mechanism which is designed to trigger the transcriptional activation of a genetic program that will enhance the autophagy, the capacity of the normal protein folding in the ER. So while this serve the purpose of cell restitution in all the cells, it is also important in terminally differentiated cells as well, such as neurons and myocytes, which does not have the regeneration capacity. So this autophagy removal of damaged cell organelles, that is a process and supports the quality control, disposal and recycling of the cells. And this autophagy, this has 5 steps, initiation, elongation, membrane closure, and degradation. Where in the initiation stage, ULK complex 1 and 2 that binds to the ATG protein and forms and initiates the autophagy process. And in the elongation, all these proteins bind, and this particular cell will get the elongated, and after engulfing all the organelles into it, the membrane will get closed, and after that it degrades the membrane or cell organelles inside the cell. This is the autophagy. Then the autophagiosome is helpful in binding membrane closure, and autolysosome degrades the membranelles present in the cell, damaged membranes or membranal organelles present in the cell. And regeneration of damaged axons. Generally, neurons in the CNS they have very limited capacity to regenerate, but axonal damage in the peripheral neurons can be repaired by axonal regeneration, which is a process that requires macrophage and squam cells. In this phase, macrophages removes debris by phagocytosis and produce cytokines and also growth factors. That cytokines and growth factors activates the squam cells, which proliferate and transdifferentiate to facilitate and support the axonal growth. So these cells, span cells, that also play an indispensable role in promoting the axonal regeneration by increasing their synthesis of cell adhesion molecules. Example, NCAM, that molecules elaborate the proteins for basement membrane reconstruction, and by producing an array of neurotropic factors like brain derived neurotropic factor, glial cell line derived neurotropic factors, and their receptors as well. So these neurons which are there in the central nervous system lose their intrinsic growth capacity postnatally. Then that before, like in the after birth, generally these the neurons which are present in the CNS that does not have the regeneration capacity, but this occurs with increased expression of some neuronal growth inhibitory couple like transcription factors, which are KLF4. This helps in increase the expression of the some neuronal growth inhibitions. So damage, as I said, like damage to the CNS neurons, that is irreversible, but in compensation for it, there is a large number of reserved nerve cells that can take over the functions of lost neurons. For example, if any neuron got lost or death happens to the neurons, there will be some reserved neurons where that takes the neurons function, and that starts the functioning of the death neurons which got death, which are died. For example of this is Parkinson's disease, which are, as I said, symptoms will not be observed, at least 80% of loss of nigrostriatal neurons. So coming to the tissue repair, in tissues with cells capable of multiplying, damage is reversed by deletion of the injured cells and regeneration of the tissue by proliferation. That is done by the apoptosis mechanism. And apoptosis supports only in constantly renewing cells in bone marrow, the respiratory and GA epithelium, because these are the cells where these are the places where the cells will get constantly renewed, and the epidermis of the skin of conditionally dividing cells as well. In this also, in this apoptosis supports the tissue repair and proliferation and regeneration of tissue. Repairs of the tissue or the tissue injury that involves the regeneration of the lost cells, reconstitution of the ECM extracellular matrix, and reintegration of the newly formed elements. So in organs like liver, kidney, lung, various types of cells will be involved in the process of tissue restoration. But non-parenchymal cells of mesenchymal origin residing in the tissue, such as resident macrophages and endothelial cells, these those migrating into the site of injury, such as monocytes, and produce factors that stimulate parenchymal cells to divide and stimulate some specialized cells like stellate cells in the liver that synthesize the ECM molecules in the tissues or the cells. And other reactions to tissue injury are in addition to the mediators that aid the replacement of lost cells and the ECM, there are some resident macrophages and endothelial cells that cells will activate the if any cell injury happens, these cells will get activated and produce other mediators that induce the ancillary reactions with uncertain benefits or harm to the tissues. So such reactions include inflammation or altered production of acute phase proteins and generalized reactions such as fever. So mechanism of adaptation. So adaptation is a mechanism where this is a kind of process where the organism develops increased tolerance to the harm itself. That involves the responses of acting to preserve or regain the biological homeostasis in the phase of increased harm. So this adaptation to toxicity may result in different biological changes that causes diminished delivery of the causative chemical to the target. So this adaptive mechanism that decreases the delivery of the target, decreases the delivery of the chemical to the target, and decrease the size or susceptibility of the target. The size of the target also will be decreased, and increased capacity of the organism to repair itself. The capacity of the organism repair mechanism will be increased. That is one of the adaptive mechanism, and mechanism to compensate for the toxic and infected dysfunction. So if there is any toxic and infected dysfunction is there, that mechanism will be compensated by in with this process. So adaptation by decreasing delivery to the target. Generally, delivery of the ultimate toxin to the target by decreasing absorption. So this mechanism helps in decreasing absorption of the chemical, so that the concentration of the chemical will be less to reach to the target, and also by this process happens by sequestration, these intracellular binding proteins that undergo sequestration process, and that binds to the chemical, so that the absorption of the chemical will be less, and the toxicity will be less. And regulation of iron absorption. So this iron regulates this intestinal absorption of iron metal, that metal ion as per the demand. So this what happens, this iron helps in regulates its own ion mechanism by, if there is any if there is a need of iron, that the absorption of the absorption of iron in the intestine will be more, and if there is no need of iron, so then the mechanism of absorption of iron will not be there. The iron will not absorb into the intestine in that step. So based on the need of the iron, the iron absorption will happen. And induction of this regulation will be done by proton coupled divalent metal transporter DMT1. This is present in the enterocytes, intestinal lumen. So induction of ferritin and metallothionein. So binding proteins, ferritin and metallothionein, these are protective against iron and cadmium respectively. Iron for ferritin and metallothionein for cadmium. So like DMT1, ferritin is also regulated by IRP1 in the phase of iron overload. So ferritin is protective as it acts to bind and remove iron ions systemically. The ferritin will comes and binds to the iron ions, and that removes the iron ions systemically based on the requirement of the iron. So induction of enzymes repairing DNA. The DNA damage response. Generally, DNA, as we all know that DNA is critical for cell survival, and damage also initiates the numerous pathways like the tumor suppressor P53, that is regarded as a guardian of the genome. That is also critical to DNA, critical in DNA repair process, and E3 ubiquitin ligase that ubiquitinates P53 and facilitates its proteasomal degradation. And then it is activated by several kinase pathways that signal in response to double stranded DNA breaks. And coming to the next process, adaptive increase in proliferative responses to promote tissue repair. Often there will be a resulting signaling cascade to initiate the tissue repair via cell replacement. In that situation, phosphatases such as PTP and PTEN, they play active role in serving the brakes, serving as brakes on growth factor receptor initiated mitogenic signaling. So these enzymes which contain reactive cysteine thiols in their active sites, they bind covalently and undergo modification covalently, and thereby and they inactivate the electrophiles, and in that manner their loss of their inhibitory activity on proliferative signaling cascade serves to amplify the intracellular signaling for mitosis and survival. And adaptation to hypoxia. The hypoxia response. Hypoxia inducible factor HIF1 is maintained at very low constitutive intracellular levels because of the continuous hydroxylation of its two proline residues, which are HIF prolyl hydroxylase. And under normal non-stress conditions, HIF1 alpha is a short-lived protein. The half-life will be very less, and HIF1 alpha kept transcriptionally inactive by hydroxylation at one of its asparginase residues by HIF aspargine hydroxylases. That prevents the interaction of HIF1 alpha transcriptional cofactors such as P300 and CRB binding. So final and irreversible actions of repair and adaptation. So the most important consequences of inappropriate repair are irreversible organ failure. As in this case, necrosis or tissue changes that advance to fibrosis or carcinogenesis. So general principles concerning failure to repair or adapt are presented here. When repair fails, what happens if the repair fails? So although the repair mechanisms operate at molecular, cellular and tissue levels, they can also fail to provide protection against injury. So in that case, the toxicity is manifested when repair of the initial injury fails, because the repair mechanisms becomes overwhelmed or exhausted or impaired. So in these cases, the failure of the repair mechanism happens. So the example for this is alkylation of DNA. DNA depletes the MGMT, and lipid peroxidation depletes the alpha tocopherol, and overproduction of oxidized or otherwise damaged proteins that also can exhaust the pool of ubiquitin. Other than the repair mechanism, if adaptation fails, what happens? So there would be moderate oxidative stress that activates the NF-KB phosphorylation pathway, or AP1 or NRF2 phosphorylation pathway, that initiates the adaptive protection. However, extensive oxidant exposure aborts these programs, because that leads to the oxidation of thiol groups in the DNA binding domain of these transcription factors. For example, tubuloglomerular feedback, which is very much important in regulating the glomerular filtration. But in acute renal injuries or tubular reabsorption, in acute renal injuries, the tubular reabsorption will get impaired, and that leads to the polyuria. That triggers a tubular glomerular feedback to further reduce glomerular blood flow and filtration. And toxicity resulting from inappropriate repair and adaptation. So failure to repair involves molecular, cellular and tissue dysfunction, and it also shows species and age related, because this happens in the based on the species. This dysfunction happens in different species, and age related difference also, and that some toxicities also involve disrepair at the isolated level. For example, hypoxia and dyspnea that develop after exposure to methyl hemoglobin forming chemicals. The amount of methyl hemoglobin produced overwhelms the capacity of the methyl hemoglobin reductase. And tissue necrosis. Progression to tissue necrosis can be interrupted by apoptosis and cell proliferation. So in this, apoptosis does this by preventing necrosis of injured cells and the consequent inflammatory responses that may cause additional injury by releasing cytotoxic mediators. So example for this is activation of Kupffer cells with administration of bacterial LPS endotoxin, which greatly exacerbates the hepatotoxicity of galactosamine. And next step is carcinogenesis. We all know that altered carcinogenesis is nothing but the altered expression of proteins that functions as proto-oncogenes and suppressor proteins. These proteins altered by mutation resulting from direct DNA damage or by altered transcription that leads to the overexpression of these proto-oncogenes or decrease the expression of tumor suppressors. That leads to the carcinogenesis. Another important consideration is that genetic or epigenetic mechanisms which are involved in the carcinogenesis. So DNA reactive or genotoxic mechanisms that bring about a qualitative change in the gene expression with expressing of an altered gene product or a mutant protein or miRNA, and with that gains or loses the activity. So coming to the pictorial representation of the carcinogenesis, here the genotoxic carcinogen leads to the DNA damage. So this DNA damage, if it gets repaired, then that leads to the cell survival. If it didn't, if it's not repaired, then that leads to the cell death. And after the DNA damage, if it's not, if it undergoes DNA replication, then that leads to the if the repair happens at the DNA replication, that leads to the cell survival or else cell death. And that next step is mutation. If it is a silent mutation, that leads to the cell survival. And if activation of oncogene, if the mutation activates the oncogene proteins, and that leads to inactivation of tumor suppressor proteins, oncogene proteins get activated and tumor suppressor protein get inactivated. That leads to the neoplastic cell transformation, that leads to the clonal expansion, that leads to the tumor. And coming to the conclusion, so a real world application of this toxicity mechanisms helps in development of the adverse outcome pathways that informs and facilitates the toxicity testing and hazard identification. So in this adverse outcome pathway, this is a systemic approach where that organizes and integrates a series of biological events that describes throughout this like described in this chapter as mechanism of toxicity that likely to lead to an adverse outcome. And this topic helps to know the basic concepts of any toxic mechanisms that identify the key events which are associated with the toxic outcomes and inform species differences in the toxicity with particular emphasis on informing the human relevance of toxicity. And the significance of this mechanism of toxicity also provides opportunities and directions to identifying the biomarkers. What could be the biomarkers of toxicity, and what could be the predictive toxicity tools, and what are the potential ways to mitigate the adverse effects. All these mechanisms and all these factors and steps can be known by knowing this mechanism of toxicity. And thank you.